Hello everyone, I'm Nahda Ahmad Rashid, actually a master's student in the Department of Fundamental and Industrial Microbiology in Istanbul University. I'm here today to share with you some of the economic values of cave bacteria. With the evolution of the industry, our environments become polluted with toxic chemicals. Avoiding such problems, replacement of synthetic materials by natural products is still the best solution. Therefore, microorganisms are the favorable source due to their continuous availability and manipulation at low cost. Research for new bioactive compounds conducts scientists to the extreme environments such as caves. Caves constitute a great microbial laboratories from where microorganisms with human welfare could be isolated. These underground environments are extreme ecosystems in which their biotic factors, especially bacteria, are found to contribute to nowadays biotechnology and industry. From drug production, bioremediation, food processing to construction, these bacteria are thought to be economically the good source of bioactive compounds used in these different processes. However, it is thought that most of the cave bacteria are uncultivated. But with the improvement of molecular-based method, microbiological investigation in caves become easier. In our actual study, we discussed the cave bacterial economy through our days biotechnology and industrial use of these bacteria as well as their biocompounds. First, the cave bacteria and biotechnology. Caves are oligotrophic environments in which the chemolithotrophic bacteria are thought to be the primary producers. The limited nutrients in the environment induce the competitivity between the living microorganisms under such stress. Bacteria secrete secondary metabolites which can be nefast for the neighbors organisms for self-protection. Caves are isolated and naturally human or animals should not be able to live in there. Therefore, no antibiotic resistance in these bacteria should not be observed. However, some of them can get the resistance genes from the excited microorganisms introduced by human or through human activities in lines. On the other hand, the resistance genes can gain naturally from the resistance phase to the secreted antimicrobial compound present in caves. The antimicrobial and anti-cancer activities of these cave bacteria. Studies around different caves in different areas in the world show the antimicrobial potential of some cave bacteria against human pathogens like MRSA and Candida albicans. It is the case of our actual run study from Yörük Düdeni in Turkey, which gives such as result as it is illustrated in my photos. We can also find some commercialized antimicrobial compounds isolated from cave bacteria, like these compounds. Also, ceramycin A and cycamycin A have been all found to have anti-cancer activities. Some cave bacteria which have negative antimicrobial activity in phenotypical experimentations have been found to harbor genes such as NRPS and PKS coding bioactive compounds. For the bioremediation potential, I will discuss through the biosorption of heavy metal, wastewater bioremediation, microplastic biodegradation potentials of cave bacteria. Some cave bacteria isolated from sulfide-rich cave like Acidothiobacillus species are sulfur oxidizer or growing at relatively low temperature. They could be favorable in bioremediation systems where mesophiles or thermophils are disadvantageous. Acidithiobacillus species isolated from the Frasessi cave system in Italy is found to be able to remove heavy metals from contaminated marine sediment. Actinobacteria like Nocardia, Streptomyces, and others from the Humboldt cave, which show resistant effects faced to toxic metals like plumb, nickel, zinc, etc., could be good candidates for the gray biotechnology like metal biosorption or bioaccumulation, since their neighbors isolated from other environments have been tested positive in biosorption, recycling, and bioaccumulation of gold, thorium, and uranium. For the wastewater bioremediation, bacteria which are calcium precipitation inducers can solve the problem of the industrial wastewater. They are mainly present in caves, below dams, and sediments. The process of microbially induced calcium precipitation is used in a diversity of fields like bioremediation of wastewater for calcium removal. During this biomineralization process, cations are incorporated into the calcite and they are immobilized within the calcium carbon structure, thereby their transportation into the environment is slowed. Cave bacteria can be also good microplastic degraders. Antibiotic producer cave bacteria such as Serratia bacillus species and Stenotrophomonas species isolated from Kashmir cave were effective in the biodegradation of polyethylene. Other cave bacterial species neighbors to microplastic degraders isolated from microplastic polluted areas can be good microplastic degraders. Also, in our laboratory, actually we run bacterial biodegradation of polypropylene microplastic, and until now we resulted with 8 bacteria isolated from different caves in Turkey, which show a decrease of the microplastic weight. Another point of biotechnology, the biopigmentation productivity potential of the cave bacteria. Pigments of microorganisms like melanin and carotenoid consist of diverse functional substances. They are acting under stress and have protection roles since they can dispense antibiotic and anti-cancer effects. Also, they protect cells against UV emission and external chemical effects. In a dark environment like caves, these pigments are more evident. Cave structures can be marked with color consisting of pigmented microorganisms. It was reported that the highest pigment producer bacteria are the Actinobacter genus Streptomyces, and these Actinobacteria are among the mostly colonizing cave walls, and pigment production in bacteria is more likely to be present in their members. These biopigments can be isolated and studied for the natural pigment use, since synthetic pigments used in many industrial and biotechnology fields such as food, allure, home-used materials, wearing clothes, and medicine are for environmental safety and healthy problems. Another importance of cave bacteria is it is for the industrial use. Enzymes are biological macromolecules produced by living organisms which bring a specific biochemical reaction acting as catalysts. Some, like of amylases, lipase, and others, are industrially important. Amylase alone counts account approximately 25% of the world enzyme market. They can be used in food processing industries such as fruit juice, also in dairy product manufacturing industry like beta-galactosidase. The proteases enzymes like acrylamidase can be also used for food product like meat fermentation. This found that 90% of bacteria isolated from Gumki cave in India have at least one enzyme potential, including lipase, amylase, and protease. Also, in a study run in our laboratory from Parsi cave bacteria through White tests show a beta-galactosidase lipase acrylamidase activities in gram-positive and gram-negative isolates. In our actual study about from the bacteria isolated from Parsi cave, also we found some of this activity, enzyme activity like gelatinase, amylase enzymes, lipase, starch hydrolysis, proteinase, and others. Another industrial field that we can use for important cave bacteria, it is in the construction. By concrete fabrication and concrete crack healing by cave bacteria through microbially induced calcite precipitation. We know that concrete is one of the major construction materials, and diverse factors like variation on the temperature and others or natural events can impact on the concrete by causing cracks on it. These cracks affect the useful life of the concrete. So to solve this problem, several mechanical and chemical methods are used, but for a safe and healthy environment, as well as for economic reasons, by concrete fabrication remains the favorable solution. This microbially induced calcite precipitation process is done through different biochemical processes such as biomineralization, urea hydrolysis, amino acid ammonification, and denitrification. But the urea hydrolysis is the most studied process, and the urea's activity shows high acid precipitation compared with the other metabolic pathway. This enzyme activity is reported in many cave isolates, especially those isolated from speleodems and karstic cave sediments. Some of them are like Acinetobacter, Steinobacillus, macrolide, Sporosarcina, Pseudomonas purida, and Sphingomonas mucosicina. The Pseudomonas purida and Sphingomonas mucosicina are two bacteria isolated from Yersinia in our laboratory. By conclude my presentation, cave bacteria are extreme microorganisms with many beneficial uses. Despite the limited study of them, they are known as source of many industrial and biotechnology tasks. They can be used in drug fabrication, bioremediation, enzyme production, and construction. Caves over the world are applied. However, most of them still unexplored. Some of them are not protected. The economic values of the cave-living microorganisms need an improvement in the cave exploration and protection techniques. Scientists, ministers, cavers, and cave managers are called to work together for this challenge. Thank you for your attention.